Hello students, welcome to LEELA'S tutorial. In last class we have discussed about callus cultures. So today let us discuss about cell suspension cultures. What do you mean by suspension cultures? The cells which are cultured in liquid media are referred as suspension cultures. Right? So how we are going to initiate that one? So coming to initiation, the first step is isolation of the cells. For going or performing the cell suspension cultures, as we know, in suspension culture it is only the single cells or the cell aggregates which are cultured, right? The cell aggregates, that means 2 or more cells, 2 or 3 cells, but not the clump of cells, right? So in cell suspension you are going to homogenize the cells, and the single cells are cultured in order to produce your secondary metabolites. Right, the plant products which are useful in pharmaceutical industry and all. Right, so here in case of bulk production, we go for these cell suspension cultures. Here first thing is isolation of the cells. So if you want a cells which has to be cultured, what you do? First you can select the explant. Right, after inoculation and incubation of the explant, you can produce the callus. And from the callus, you can go for homogenization, right? We have discussed in last class, right? You can go for the homogenization and simply suspend all these in the liquid media and go for the suspension culture, right? Then you go for 2 or 3 passages. After that passages, you can go for mass production. Okay? So here, either you can take the callus and homogenize or disaggregate the cells present in the callus and collect the single cells. Otherwise, directly from the plant organ, either the shoot system or the root system, whatever explant you are selecting, from that plant organ, either by mechanical method. Mechanical method, simply taking the mortar and pestle and grinding those explants, either the leaf or the stem or the root. Right, so simply mechanical method, by means of mortar and pestle you are going to grind it, and then the extract whatever you are getting, that you are going to centrifuge and collect the live cells. Okay, it is a crude method. Otherwise you can go for the enzymatic separation. So in case of enzymatic separation, you can make use of enzymes such as macerozyme, pectinase, cellulase, right? So here, by these enzymes you can obtain single cells. Then the collected single cells can be suspended in plant tissue culture media, and you can culture these suspension cultures. Okay. So here the next step is growth pattern. So once you are producing, you have inoculated the single cells in the conical flask or any liquid media. What is there after inoculation? It is the incubation. Why you incubate? In order to allow the cells to grow and develop. Right? They must increase in number. Right? The cells must increase in number, and also they must increase in size. Okay? So growth and development must be there, and which must be monitored simply by taking the turbidity levels in the conical flask. We can plot the graph. This is the growth curve or the growth pattern which is happening inside the conical flask after inoculation and incubation. So here you can see the pattern. So here y-axis is number of cells and the x-axis is culture time. Alright. So here when you inoculate into a conical flask the single cells in the plant tissue culture media, first it is the lag phase. Lag phase where there is no immediate increase in number of cells. That means you have inoculated in a fresh media. Now the cells has to get adjusted to the new environment. Right? When they get adjusted to the new environment, they can take up that nutrients from that media and start cell division and development. Right? So at this stage, they are getting adjusted to the new environment. Okay? So then here you don't have any quick growth. Then you can see as the time is increasing, the incubation period is increasing, here the cells slowly start increasing. And now this increasing is referred as log phase or exponential phase, because they grow at exponential rate 2 to the to power n rate, right? So there is a rapid increase in the number of cells and also the size of the cell. As a result, you can see the graph has increased. The number of cells has increased within a short time. Okay, so now this is called as log phase. Then as the media is getting depleted, now we are growing the cells or culturing the cells in some media. So as the cells are dividing and growing, there may be depletion of nutrients in that media. Due to that depletion or due to some other factors, there may be cell growth has stopped now. The cell growth or the number of cells which are increasing has stopped, and now only the cell development is observed. Cell development means the cell number is constant but the size of the cells is increasing. So once the cell size is increasing, now these are the mature cells where we can produce secondary metabolites, right? So here the bioactive compounds or the secondary metabolites or the plant products which are used in pharmaceutical industry can be produced at this stage of the plant cell. Okay, if you are not maintaining here properly, then they may enter into the decline phase or the death phase, where the cells are gradually decreasing in number. That means the cells are dying, right? So due to depletion of the nutrients or due to some other environmental factors. So you have to maintain the stationary phase of the cells in order to produce your bioproducts. Okay, so then next one is maintenance. So how you are going to maintain? Once you are inoculating them in the new media, right, their aeration must be present, right? So the continuous agitation must be present by using your magnetic stirrer or shaker incubator or orbital shaker or any other incubators or any other mechanism can be used in order to move the conical flask or move the containers, so that there will be proper mixing of the media to the cells and also aeration will be proper. So like that you are going to maintain the cells and go for the bulk production. So here the bulk production again differentiated into two types: batch culture and continuous culture. So this is an open type of culturing process, whereas the continuous is a closed type of culturing process. Okay, let us see diagrammatically the first one, the batch culture. In the batch culture, the name itself is implying that only a batch or one set of production will be done. Okay, so here in the batch culture, this is the bioreactor, right? If you see the bioreactor or the fermenter, here you have the inlet and the outlet, and here this is the agitator. Okay, so that it mixes all the suspended cells with the liquid media. Okay, so it will be rotating very gently. Okay, from 80 to 200 RPM. That is rotations per minute. So it will not rupture the cells, but it simply agitates or mixes all the cells with the liquid media. Then you have the sparger which releases the oxygen or air into the fermenter, right? So then you have the sensor where it will check for the pH, variation in the pH or any toxins released, the toxic waste which are released by the plant cells, everything will be sensed there. The temperature, everything. If the temperature is increasing, the cooling jackets will be present in order to stabilize the temperature. And you have here the baffles. So baffles, in order to avoid the vertex formation, as it is rotating, there may be formation of vertex. In order to avoid that vertex formation, you have the baffles inside. Okay, so this is a batch fermentation where you will be adding the media once, right? Media will be added once in the bioreactor. Then the cells are allowed to grow, right? The cells are allowed to grow and maintain the stationary phase so that they are producing your secondary metabolites or bioactive compounds. Okay, so once the complete fermentation of complete process is completed, right? Once the production is completed, what you will do, you will release out or collect all the media along with your byproducts and you go for the downstream process. That is purification of your product, right? That we'll see later in detail. So now now you have collected everything out, right? So again it is the cleaning of your bioreactor and sterilizing all the valves, inlet, outlet, and completely you will be sterilizing by means of steam sterilization process, right? Then again another batch or another set of media you will be adding. Again same process, collect it. Again sterilize it, clean it, right? Again another batch. So like that, this must be occurred in batches. It is not a continuous process. You are going to get the product, again start a fresh batch, again collect the product and again next batch. So it is referred as batch culture or batch production of your secondary metabolites. Then it is the second one, continuous culture. So in case of continuous culture, there is no need of sterilizing in between. So once you sterilize your tank, valves, everything, okay, once it is sterilized, you will start with the production. So here the fermenter or the bioreactor is connected with the sterile media tank. So the media will be continuously passing into the main bioreactor where the cells are present. Here you have again the baffles, agitator, sparger, everything, right? Here pH, temperature probes, everything is present. So here continuously media will be added and the production is continuous. And here you have the light source and photocell in order to check for the turbidity. Okay, so here you are continuously adding the media and continuously you are taking out the media along with your byproducts. There is no intermittent addition of any, intermittent sterilization process. Okay, so continuously you are adding and continuously you are taking out after a certain period. It is very slow process, but it is a continuous process. Okay, so the time, very slow movement of the inlet and slow movement of the outlet will be there. Is that clear? It is the difference between the batch and continuous. In batch, you are only one set, you are adding the media and after completion of the process, you are collecting it out. And again you will start next batch. Whereas here, it is a continuous process where you are adding the media and collecting the media continuously, right? The process is never stopped. It is a continuous process. It may take up to 6 months or till the contamination is observed, right? So here you have the sensors for checking the toxics of waste or pH, variation in the pH, temperature or any other disturbances. In that case, they will stop and again sterilize. Is that clear? Otherwise, it is a continuous process.